Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all the participants of International Conference on Environmental and Energy Sustainabilities. And thank you for the committee of ICES for hosting this conference. My name is Rioneli Ghaudenson, and allow me to present our paper titled Effectiveness of Groundwater Boiling as Household Water Treatment in Metro and Bekasi Cities, Indonesia. These are the outline of today's presentation. We start with introduction. This is the research background. There is a consistent national shortage in pipe water distribution system in Indonesia. In 2020, the service coverage persisted at 20.29%. And as a consequence, most households tend to use improvise with other alternatives, including groundwater in the form of dug wells and boreholes. And unfortunately, groundwater in Indonesia demonstrate a high contamination risk from solid waste, animal extraction, and a polluted environment. Therefore, to counteract this negative effect, boiling is extensively applied with government support over the past decades. However, improper boiling practices is not able to completely eliminate all pathogens. And apart from water source, recontamination possibly occurs after treatment due to inadequate storage. And this is the research objectives. Firstly is to assess microbiological effectiveness of boiling as a domestically preferred household water treatment method. And the second is to analyze the influencing factors of water quality change from the source to the point of use. This is the research methods. The study was conducted over a 4-week period in 2 cities, Bekasi and Metro, between February and March, and October and November in 2020, respectively. 3 peri-urban districts in Bekasi and Vive in Metro were selected as study location. This location was selected due to high poverty rate, high population density, reliance on groundwater, and limited access to pipe utility supply. The total household flow rate was 563. But the point of use water sampling was only done in 176 households. And of this 176 households that uses groundwater for drinking was 116 households. And the focus of this study covers the households using groundwater and boiling as a primary water treatment method. And so in Metro, the total sample is 65 households, while in Bekasi, it's 51 households. For the household data collection, we use an online questionnaire developed with Qualtrics. The survey was composed in English and translated to Bahasa Indonesia. Local interviewers from Bekasi and Metro were adequately trained and equipped with sufficient resources for water sampling. In Metro City, strict health protocols were observed due to COVID-19 pandemic. For the water sample collection, water samples are taken from tap and glass and stored in water bag. Then we measure the temperature, pH, and TDS, and we store them in a cooler bag to be analyzed in field lab. And in the field lab, we prepare 100 milliliters of water sample, and we add the colored 18 reagent, and we pour them to a quantity tray. We seal them and incubate them for 18 to 22 hours. And then we quantify E. coli based on the fluorescence. For data analysis, the questionnaire and laboratory data were analyzed using Microsoft Excel and Stata. Log 10 was applied to transform E. coli NPN count. And water quality analysis involved the use of binary variables to indicate the presence or absence of E. coli. And as a result of abnormal data distribution, non-parametric statistical tests were employed in this research. And for the results and discussions, this is the respondent characteristics. From a total of 116 respondents, the majority of households in Bekasi and Metro employed boreholes as a primary drinking water source. And in total, 41 of the 116 or 35% point of view samples are contaminated with E. coli. About 96% of households in both cities employ boiling as a primary treatment method. For residents using boiling method to treat the groundwater, 36% were found to be contaminated. The most common water container used are jugs. Most respondents reported using closed water container, but 36% of the samples are contaminated. Samples which were stored in less than a day before consumption was found to be least contaminated. For the boiling effectiveness, Wilcoxon site rank test showed a statistically significant difference between source and boil samples. This means that there is a significant E. coli reduction before and after boiling. However, the E. coli reduction value for both cities, which was 70.5%, is less than previous studies in Guatemala, which was 82.6%, in India, 99%, and in Vietnam, which was 96%. Which indicates inadequate boiling practices in Bekasi and metro cities. And for the water quality change, this is the water quality change, which was categorized in risk level based on WHO risk level, ranging from low, medium, high to very high based on the E. coli concentration. Overall, we can see that the point of view samples had a lower risk compared to the source samples. 46% of the source samples use an improvement in risk category, indicating the success of boiling as a treatment method. 42% sees no change in risk level, and 11% of the source samples sees a deterioration in risk level. An extreme case that we found was in the source sample that has a low risk level. At the point of use, it jumped to a very high risk level. So there is an indication of recontamination in there. 11% of the deteriorated cell samples could be attributed to recontamination in water containers and drinking cups after boiling. However, the experiment strength test for different water storage and cups with presence or absence of E. coli indicates no statistically significant correlation with p value of 0.43 and 0.32 respectively. In conclusion, boiling treatment method in Bekasi Metro demonstrated an improvement in water quality. However, it failed to completely eliminate the microbial risk with only 70.5% reduction of E. coli. Water quality change analysis revealed an improved risk category in 46% of the samples after boiling, further assuring the effectiveness of boiling in peri-urban settings. However, in 11% of cases, water quality deteriorated between source and point of use. This finding may be indicative of inadequate boiling processes or a possible recontamination while water is being stored. However, the study observed no statistically significant correlation between type of water storage vessel and the presence of or absence of E. coli. It is the suggestions. Future researches is needed to understand the mechanism that results in fecal contamination at source and point of use, and also to better pinpoint why boiling practices in some households are inadequate, especially in Indonesia. And that's all from me. Thank you for your attention.